is a rice field in Taiwan after an insect attack.
This is a coffee tree in Uganda after an insect attack.
This is a grape tree in Spain after an insect attack.
This is a soybean field in Argentina after an insect attack.
Harmful insects are causing grave damage to our lives.
Each year, agricultural loss caused by insect damage alone is $470 billion.
20% of the annual crop production is destroyed by harmful insects.
If some of this food can be saved from insect damage, it can be used to feed the more than 3 billion people who are starving around the world every single day.
Insect damage has always been a problem, and it has continued to be a problem, even with the advance in technology nowadays.
This problem has to be solved, and people have never stopped trying.
However, our current solution is pesticide. 3 billion tons of pesticides is being applied each year on crops.
Pesticides can cause severe health problems such as birth defect, nerve damage, and cancer. It is also greatly damaging our environment in all its different forms of devastation. We are the 2014 NCTU Formosa iGEM team.
We need a Chen.
Good morning, ladies and gentlemen. We are Team NCTU Formosa, and today we are here to give you a briefing on our project this year, Operation Debug. I am Supervisory Special Agent Rachel Lee.
And I am Supervisory Special Agent Nick Shaw.
We are from the Harmful Insect Elimination Force, and our headquarters is based in National Jiao University, Hsinchu, Taiwan. As our name suggests, our task force is dedicated to the solving of insect damage.
Hundreds of thousands of hectares of production is wasted around the world every year.
Currently, our solution is pesticides, yet we know it is a very lousy solution.
$9.6 billion are lost on damage of pesticides annually.
Our goal is to resolve insect damage without the terrible side effects of pesticides. Oh, boss is calling.
Oh, boss, oh my god, I'm not ready yet. Oh, what?
Good morning, Chris.
Status update.
In a relationship.
No, you're single, come on.
Agents, status update on Operation Debug.
Yes, sir. As we know, in nature, opposites attract. So we are using this to our advantage. We will manipulate a female moth and allow it to attract more of the very same kind of male moths. At this point of our operation, we have already acquired our target.
In this operation, we are mainly targeting Oriental lympho moths. Its main area of activity is tropical islands, and a major part of the damage is done in Taiwan.
This insect is highly dangerous and is a serious threat to our nation's security. It feeds on over 40 families of plants, which basically includes almost everything.
It is a danger to every crop.
And we are hunting it down.
So, what's your plan?
Yeah, we are planning to have I was planning to have honeymoon previously.
Use that on your personal leave this year. I'm the one who's going on honeymoon. So, what's your plan?
Oh yes, sir. The honeymoon is...
Nick, we need the technical plan of operation. Sir, I apologize for my partner's behavior. Currently, as we know, the most commonly used method to attract and capture insects is the pheromone trap. As we know, pheromones are the insect's hormones originally used for mating and certain other purposes. What's so special about it is that it can specifically attract insects. So, originally, our plan was to biologically synthesize pheromones to replace the currently in use chemically synthesized ones. But we found the biosynthetic pathway of pheromones way too complicated for E. coli to carry out. It is practically impossible, so this mission plan was aborted. We started looking for a plan B, and we decided on infiltration. We are sending in an undercover agent, P-band, whose full name is pheromone biosynthesis activating neuropeptide. Agent P-band will train our targets insects against themselves, prompt them to start making pheromones for us, and in turn attract more of the very same kind of insects for us. So how exactly exactly is this going to work? Here we have the pheromone gland of our target insect. And once the P-band binds with the receptor on the gland, pheromone baths and thesis will be activated, and then the attraction effect will be started. So forget this complicated reaction. I mean, let's just let the insect do the job for us, because after all, they are naturally designed to do so. Now, I would like to give more information as to why we chose iGEM P-band for this operation. A lot of it has to do with its identity. It is a simple small peptide. Its being a peptide means that it can be easily made by E. coli simply through expression. And the fact that it is smaller than 150 base pairs is another plus because it wouldn't place any stress on the E. coli for expressing it. As a peptide, yet another advantage is that it wouldn't be easily degraded because it's already in a very, very simple form. Now, the most important part is it is specific. P-band has the same species specificity as pheromones. One kind of P-band can only react with one kind of insect who will in turn release its own pheromones and attract more of the very same kind of insects. So if we allow 3 different kinds of insects come in contact with the same kind of P-band, what will happen is only the corresponding kind would react. So in this case, we're using the moth's pheromone. So nothing will happen to the mosquito or the ants. Only the moth will react with the moth's P-band, release moth pheromones, and attract more moths. Okay, so now I want to talk about biobrick assembly. As you can see, it is fairly simple. We use a constitutive promoter, combine our ribosome binding site, and the DNA sequence of our P-band. We named this P-band biobrick P-band SL, taking the first letters of the Latin name of Oriental leafworm moth. So, Bob, what do you think so far?
Very well. So, where are you in your plan now?
Okay, to confirm that E. coli can actually make P-band, we first did a PCR test with the forward primer before the promoter and the reverse primer after the P-band sequence. Theoretically, the length should be around 600 base pairs, and our result is a clear single band right between 750 base pairs and 500 base pairs. So this informs us of the first step of our success. We then really wanted to see the actual expression of our peptide. So we did an SDS page gel, and again, compared to loading control, we have a clear band right at 3.7 kilodalton, the correct length for our P-band. We then constructed another biobrick assembly tagging a BFP gene as a reporter gene. The result is that our E. coli gives off blue fluorescence, meaning again that P-band is indeed successfully expressed. But this is not the only purpose of tagging a BFP gene. We can also use the fluorescence intensity to find the best time point to gain the maximum yield of our P-band. Now that we know E. coli can actually make P-band, what we want to do is we want to take this P-band and let it make pheromones and allow the actual attraction to begin. So you can see from this diagram that P-band binds with a G protein coupled receptor and once activated will initiate the calcium pump and activate the entire biosynthetic reaction of pheromones. So how do we conquer the cell membrane barrier of E. coli and use the P-band made inside the E. coli? We did something very simple. First of all, we poured out the liquid broth and then we added some buffer and then we just mash this entire solution. So we break the cells, allowing the P-band inside to come out and into the solution. And then we dilute it to our desired concentration, take it to autoclave, and gain an E. coli-free, biologically safe P-band solution. We then soak cotton into this solution and sprinkle sugar on the cotton. When our target insects come to eat the sugar on the cotton, they will directly come in contact with the P-band soaked on the cotton. Now, on the left, we have a female moth ingesting a sugar-only solution with no P-band inside. And on the right, we have the same kind of female moth under same conditions ingesting the P-band solution we made. You can clearly see that there is no change in the behavior before or after the ingestion for the moth on the left. But on the right, the moth is frantically flapping its wings. This is a very, very, very important milestone of our project. This means that our P-band works. So from this point on, this female moth is officially sexually stimulated and it is giving off pheromones to attract more of its male counterparts.
Wonderful! And what weapon are you going to use for this operation?
Well, a large caliber handgun wouldn't do it. A Glock would have too much recoil for accurate aim. A Beretta, a SIG. I don't think any of our currently existing weapons would do the job. What do we do, Nick? Boss is gonna kill us.
Okay, so we have to design our own weapon. Say hi to my baby. Just as we previously mentioned, we hope to not only establish a new solution to insect damage currently happening all over the world, but also apply that solution to agriculture and even everyday life. For example, your background. You can see on slide that our device is like a pyramid. We choose accurate panels as our material in order to ensure it is strength enough to support our device and not easily destroyed by pests. Now, let me introduce the detailed structure of our device for you. It can be divided into several parts. First, the base. At the center, we design a rest neck structure to hold our plate, and another rest structure for the inner shell to be clicked on. Next, the outer shell. It is formed by 4 equilateral triangles. The reason why we choose the pyramid to be our shape is that it facilitates draining of water and snow and prevent clogging. This allows our device to function normally in any weather condition. We also design a pyramid with its top part chopped off as an inner shell. We continue to use the pyramid shape not simply for overall design and consistency, but also create a smoothly inclined duct for the target harmful insect to easily enter our device and unable to escape. We press turn LEDs on the inner surface of the inner shell. They are powered by a battery will be positioned here. This is not only for making our device look cool at night, but plays a crucial role of attracting the first target insect into our device and make film for us. So now, it's time to tell you how we use our device. To make insect help us, first of all, we have to make insect come to us. To do so, we will use light to attract them into our device. Once inside the device, they will be led to our P-band by the sugar we coated our P-band soft cutter. Until this point, there is no specificity in the attraction. However, once the target insect enters our device and ingests its own P-band, it will release film. Then more of the target insect will be attracted into our device. So now, our target harmful insect has already released its own film. So even if we turn off the light, we can still attract the target insect into our device. And we did an experiment using our device to see the attraction effect of blue light and P-band. Here we have a result. You can clearly see that blue light alone can attract much fewer moths than the combination of blue light and P-band which our system uses. So now, we have a video of our device working its magic. Take a look.
In this experiment, we use a chamber that simulates the natural environment. We place vegetable leaves and flowers, which are both appealing food sources for moths. Inside the chamber, there are also around 150 male moths planted for experimental uses.
Now, we press the female mouse into the speaker and it will ingest P-band. After everything is set, we press the pyramid into a chamber to begin our experiment.
As you can clearly see, that originally all of the male moths were just wandering around and minding their business, not really doing anything inside the chamber.
But after our system takes effect, to our excitement, they were all attracted into our device.
After 11 hours, we successfully captured dozens of harmful insects into our device, and none of the unrelated species were affected at all. With our new method, we are trying to start an agricultural revolution. The concept is eco-friendly, easy to use, low cost, and fashionable.
Our device is part of this revolution.
Not only does it effectively attract and capture harmful insects, but it will become the must-have garden decoration. It will light up your garden, or your farm, or wherever you choose to hang your device like a beacon in the night.
This is our device, and it is magically beautiful.
Developed in style, we are your best choice.
temperatures. For example, if it is 22 degrees today, farmers can look at our modeling result and choose blue light, which will give them the best result of insect traction. But as you can see from the result, blue light can attract much more moths than any other light in no temperature condition. So we use blue light in our device.
Now that our biggest threat is eliminated, we are taking a step back to help the rest of the world continue to fight its evils. You can see that all of these insects are also currently wreaking havoc on our world. So we constructed biobricks with their P-bands as well. With a little bit of effort, all of their problems can be solved by our system and our P-bands. Now I would like to proudly present to you the results of all of these P-band biobricks. First of all, the PCR results show that we successfully cloned all of the P-band genes onto their separate backbones. And the SDS pagetail results show that there is a clear band at each of the separately correct positions. So this means that all of the P-bands are indeed successfully expressed. The BFP tagging result tells us that all of the E. coli strains give off blue fluorescence, which again means that the P-bands are successfully expressed, and we can use the fluorescence intensity to find the different yields at different time points.
As fine as your work is, our agents alone cannot accomplish this task. A hero can be anyone. We just have to find it. And this is done through the practice amongst humans.
Yes, sir. Through our human practice, we not only took our project to the industry, we also educate people, be it farmers or students, of the importance of protecting our environment. Our human practice can be separated into 3 criteria. First, also the most significant factor of our operation is the people who support us. We cooperate with some organic farms to take our operation out of the lab and into reality. We now need to call our target insect at this farm to complete our testing. We also provide them with our device so that they can use it and give us some feedback. Second, we're the mini meetups with our iGEM team. This summer, our team held the 2014 iGEM conference, inviting 22 teams and over 150 people in Taiwan and China to attend. In this meetup, we shared our project with each other and exchanged thoughts and ideas in hopes of perfecting our project and also getting to know more iGEMers around the world. Third, we promote iGEM with a passion. This summer, our team held a biocamp to introduce iGEM to senior high school students. In addition, we talked to farmers at a green farmers market fair about our project. With their everyday farming experience, they gave us a lot of useful advice. They are amazed by the concept of iGEM as well. They were very excited to cooperate and help iGEM teams bring better life to people.
So we've talked about a lot of things, but what exactly have we achieved this year? We constructed biobricks, of course. We converted 9 P-bands into standard biobrick parts, and for each of the P-bands, we have a composite part with the promoter and the RBS, and another composite part with the BFP tagging function. So all in all, we have 27 new biobricks constructed this year. Secondly, we successfully resolved insect damage with our new system, and we crafted a new device allowing the application of this system. Third, we didn't just stay in the lab and say, ooh, everything works perfectly on Hyper. We walked out of the lab and we talked to the industry, and we took our system into the real-life agricultural environment. Also, through this collaboration, we talked to farmers and high schoolers about the importance of eco-friendliness and iGEM. Fourth, we held an iGEM conference inviting 22 teams from both China and Taiwan, and over 150 people from many different countries. We also met up with many other iGEM teams, and all of it has been a memorable journey. Last, but definitely not least, we helped TCU Taiwan iGEM team with their modeling, and our collaboration made our friendship very strong. Now, I would like to give thanks to all of our instructors and alumni sponsors for their support, and also every single member of this team. Thank you for making NCTU Formosa the team we are today. This is the end of our presentation.
Thank you.
放班级老师照片。哇，这个效果特别好。啊，我。
So let's give another round of applause for all of our undergraduate finalists. At this time, we would like to ask one team member with their computer and their presentation from each of the overgraduate presenters to come up to the front so we can work out getting your presentation set up. And for everyone else, we're going to take a 15-minute break. So the three overgrad teams, once again, TU Darmstadt, UC Davis, and Wageningen. Please come forward.